Before we get into details of auditory neuropathy, we need to understand the differences between ABR and OAEs. For example, if we travel back in time to the late 90s and early 2000s, during those times OAEs just became available, and many people are using ABRs or OAEs for hearing screening for newborns, for example. And of course, there are pros and cons for each approach. And if we look at our ABR first, say ABR sound comes into the auditory system, and the nerves will fire action potentials. So ABRs are basically screens for neurosynchrony of the auditory pathway. And the good thing is that is because the advantage of ABR is it will be able to detect the presence of auditory neuropathy, because auditory neuropathy is a disorder that lack of neurosynchrony. We'll get to details in later slides. For now, just know that auditory neuropathy is a lack of neurosynchrony. So if we use ABR as a screening tool or as a diagnostic tool, then ABR will be able to screen neurosynchrony for the auditory neuropathy. As a result, ABR will be able to detect the presence of auditory neuropathy. However, now as you have known that ABR has limitations in different spatial configurations, you have to do specific work and set up different paradigm to get that work. If you are thinking about the hearing screening for newborns, for that purpose, people use clicks only, then the ABRs is unable to provide low frequency information, and also only a very rough estimate between 2 and 4,000 Hz average. So when we use ABRs, there are pros and cons. The advantage is we will be able to detect auditory neuropathy. The downside is we are unable to detect a specific audiometric configuration. The other option is to use OAEs. OAEs, as you know, is sound comes into the auditory system, and the outer hair cells will produce echoes coming back to be recorded. So OAEs are frequency specific, because you can have OAE presented at different frequencies or as click that contains all frequency components. So long story short is OAE is a frequency specific screening. However, it screens only primarily the outer hair cell function, and of course the outer external ear, tympanic membrane, and middle ear functions must be normal as well. So OAEs provides frequency-specific screening for the cochlea, for the middle ear functions. However, it will miss the auditory neuropathy. So the pros and cons for OAEs and ABRs are just the opposite. ABR is screening for neurosynchrony and is good to detect auditory neuropathy, but it's bad for specific audiometric configuration. OAEs are the opposite. OAEs provide frequency-specific screening for the cochlea and middle ear cavity. It's able to identify different audiometric configuration. However, it will not be able to detect auditory neuropathy. So you now know, if you care about auditory neuropathy, you would use ABR as a screening tool. However, if you care more about frequency-specific information, or you care more about the specific audiometric configuration, then you will use OAE as a screening tool. This is from the standpoint of auditory neuropathy. I mean, there are many other factors that will affect whether you will use ABR machine or OAE machine for hearing screening. That's another huge topic to discuss. For now, we will focus on their relations to auditory neuropathy. For auditory neuropathy, if you use ABRs, you will be able to detect auditory neuropathy. On the other hand, if you use OAE for hearing screening, you will miss auditory neuropathy. that's kind of the opening statement for the hearing screening that will lead to details of auditory neuropathy. Before we talked about auditory neuropathy, the mechanisms and the pathology of auditory neuropathy, we need to do a brief review on the anatomy of the organ of Corti. Specifically, we need to know the, say, outer hair cells here and inner hair cell here, and we have a lot of different nerve endings or nerve fibers, and then coming to the auditory nerve, and these many circles are the auditory ganglion cells, or people say spiral ganglion cells. These are the cell bodies of the auditory nerve. And then after that is the postsynaptic axons for these auditory nerves. So this is kind of a zoom in schematic for the organ of Corti and auditory nerve. So the purpose of this figure is to show many things. One is show outer hair cells, and then show inner hair cells, and also show all the different kinds of nerve fibers or nerve endings and synapses. So now we will focus on the synapses for the inner hair cells, because that's the beginning of the ABR. So when sound comes in, it will excite the inner hair cells, and the inner hair cell will have a synapse, will have a lot of synapses to the nerve endings, usually at the bottom of the hair cell, of the inner hair cell. And then these nerve endings will then come to the cell body of the auditory nerve, and then there are also nerve fibers after the cell body. So as you can see, the synapse is at the bottom of the inner hair cell, between the bottom of the inner hair cell and the dendrite of the auditory nerve. This portion is the dendrite, and then cell body here, and then the axon after that. So the nerve endings in this area, some are myelinated, some are unmyelinated, and the cell bodies here, and cell bodies are myelinated. This is a very special feature of auditory nerves. The figure here does not show that myelination sheath very clearly, but just so you know that the cell bodies of the auditory spiral ganglion cells are myelinated. They are, so in other words, the myelin sheath wraps around the cell body of the auditory nerves, and then after that is the axons for the auditory nerve fiber. So all these different kinds of nerve fibers, some with myelination, some without myelination, and the cell body is also myelinated. And the axons mostly are myelinated, but some still are not myelinated. So all these different things and the synapses, all these points may create delays in the transduction process and the propagation of action potentials. So any point along this pathway, if any point creates a certain amount of delay, then neurosynchrony will be affected, will be disrupted. So this is the reason I show this figure, because I am using these figures to emphasize there are many different locations: the inner hair cell itself, the synapses, the nerve fibers of the auditory spiral ganglion cells, the cell body of the spiral ganglion cell, and so on and so forth. There are many different areas or joints or points that may create delays, different amount of delays, and that will disrupt neurosynchrony. Here is another schematic to show the mechanisms, possible mechanisms of auditory neuropathy. So here on top here is an inner hair cell. The hair cell itself could create delay. And then we have a lot of synapses between the inner hair cell and the auditory spiral ganglion neurons. SGN stands for spiral ganglion neurons for the auditory nerve. They are the auditory nerve. It's just for the scientific purposes, many people call auditory nerve neurons as spiral ganglion neurons. So the synapses could create delays. The nerve endings, the dendrites here, could create delay. The cell bodies here, because cell bodies here are also myelinated. Imagine when you have action potential trying to jump through this myelin sheath. It will be more difficult to jump through the section of myelin sheath that contains the cell body, because the section of myelin sheath that contains the cell body is thicker and wider. It will be more difficult for action potential to jump over the section that contains cell body, and that could create different amount of delay. And then the axon portion, and then the synapses between the spiral ganglion neurons and the cochlear nuclei neurons, and even the axons and synapses after that, up to the brainstem area and up to the cortex, and then even the efferent system coming back to regulate and control the entire auditory system. Those efferent systems coming from a top-down mechanism can also create additional delays. So again, there are many different mechanisms and reasons that can create different many different reasons to interrupt a neurosynchrony. But the end result will be similar as shown on this figure. The left-hand side column here shows a normal ABR, or let's say a normal participant. In this case, they draw three neurons as an example, three action potentials coming from three spiral ganglion neurons. Say we have three spiral ganglion neurons here, and that gives us three corresponding action potentials. And in normal participants, the three action potentials should be in sync. They should be synchronized. As a result, when we add the three together, it gives us a good, nice looking summed waveform. Now, if we look at the second column, if there is a delay, if there are delays, however, the delays are constant. In other words, it's a homogeneous delay. I mean, say each action potential got delayed, let's say 1 millisecond, for example. When the delay is constant, it's a fixed amount, as shown here. As they are all delayed with the same amount, say 1 millisecond, the action potentials are still aligned quite well because the delay is constant. All action potentials got delayed, say 1 millisecond. When you add them all together, they still give you a very nice looking waveform. It's just delayed for 1 millisecond as an example. So if the delays are constant, it's not really a big issue because it's just the waveform, the recorded waveform will be just delayed with that constant amount. So for that, we still will be able to see, responses in the ABRs. It's just the time will be different, but the response will still be present and identifiable just with a longer delay. So that's the consequence with a constant homogeneous delay. The main problem is when the delays are variable. When the delay is different among neurons, and that will create a big problem because in this situation, and as shown on this case, the three neurons here, they have different amount of delays. As a result, the three action potentials do not align together. the three action potentials are not aligned perfectly. There are some delays, different amount of delays. So when you add them all together, the sum, the waveform got compromised because they are not aligned well. As a result, they may cancel out each other partially. As a result, the waveform get compromised quite a bit. Not to mention if you have some damage to the spiral ganglion neurons, for example. So the fourth condition is giving a condition that some spiral ganglion neurons are non-functional, say they are dead, or for any reason they are non-functional. In this example, let's say 2 out of 3 spiral ganglion neurons are dead or non-functional. Only one will respond. And for this only one neuron that's responding, there's no delay. It's that surviving neuron is healthy. Then you just get one little action potential here. When you add them together, you still get only one very little amplitude here. And if you have variable delays, and also on top of that, you have some loss of spiral ganglion neurons, as shown in the last condition here. You only have say one neuron surviving. The other two are not functional or dead. And this only surviving neuron also provides some amount of delay. In this case, not just you have one small action potential, you also have the response is actually minimized or disappear. It's very, very small. So this is kind of the illustration of mechanisms of auditory dyssynchrony, or we should say neural dyssynchrony in the auditory system. So for whatever reason, as long as the neural synchrony got disrupted, it could be different amount of delays, could be the loss of spiral ganglion neurons, could be constant delays, could be a combination of any of these reasons. The bottom line really is, as long as the neural synchrony got disrupted, ABR may just be highly compromised. ABR waveforms may be highly degraded or disappear. So this is the reason that when we lose neural synchrony, ABR waveforms will be highly degraded or simply disappear. And that is what we see in patients with auditory neuropathy. So here is a possible clinical profile for patients with auditory neuropathy. The bottom line really is you have evidence of normal outer hair cells in a child, infant, or a person or a patient. However, this child, infant, patient, or individual have abnormal ABRs and or abnormal behavior responses. So this happened in the early days when people, in the late 90s and early 2000s, when people have the ability to do OAEs. Then at that point, when they combine OAEs and ABRs, they found, hey, we found a subgroup of patients who have OAEs, but their ABRs are abnormal. And that's weird. So at that time, that's weird. So they have OAEs. As you now know, OAEs indicates outer hair cell functions. So normal OAEs indicates normal outer hair cell functions. So these patients, their outer hair cell functions are normal. And their middle ear functions are also normal. However, they have no acoustic reflexes. It's strange. If you present sound to try to evoke acoustic reflexes, you get no acoustic reflexes. And also, when you try to record ABRs, the ABRs are either totally absent, or you can only see the very early waves. The ABR waveforms are highly degraded. Either they are totally gone or highly degraded. It's strange. And for the behavior thresholds, is even strange. the patient could have normal hearing thresholds. I mean, normal, like normal, nothing wrong with any behavioral thresholds. The patient can also have profound, severe to profound hearing loss in terms of pure tone audiogram or speech perception. So behavioral thresholds and performance are just all over the place. It could be anywhere. And they also found that many children have the evidence of other peripheral neuropathies. So it seems like these kind of patients, auditory neuropathy patients, tend to have other peripheral neuropathies as well. And also, if newborns or infants, they had hyperbilirubinemia at birth, they have higher chance of developing auditory neuropathy either right after birth or later in time. So this is the clinical profile for patients with auditory neuropathy. It's kind of it's kind of a mess because there are not many things that are for sure. It's a lot of different variations in there. One thing for sure is you likely will have LOAEs, and you don't have acoustic reflex, and the ABR is absent or only the early waves. And so this clinical profile for auditory neuropathy is quite messy, and that probably is the reason people call it auditory neuropathy spectrum disorders, because it may as well include a spectrum of different disorders. It's just for now we are unable to differentiate different specific pathology. So say you have a newborn infant who passed OAEs but failed ABR hearing screening in the newborn center. At that moment, you really don't know what was the actual mechanism for that specific infant. You don't. Even when they grow up, you still don't know. Most of the time, we still are unable to pinpoint the site of lesion. The chance is those neurosynchrony are a result of neural abnormalities. So that's what we think, and it could be the neurons, it could be the synapses, could be the dendrites, could be the axons, it could be the hair cells. So it could be a lot of things we are not sure yet. And also, most patients with auditory neuropathy spectrum disorders, they show bilateral symptoms. They show symptoms in both ears. This is because, as you can see from this, all these mechanisms seem to be symmetric. It's not something you get hit by a baseball on the right temporal bone, and it's not that. It's something that seems to be systematic for the entire human body. So, as you know, for neurosynchrony thing, if you do cochlear implantation, the electrical stimulation is a much more powerful stimulation than acoustic stimulation. So given electrical stimulation is a powerful stimulation, when you zap the nerves, all the nerve fibers will fire action potentials essentially at the same time. So if you use electrical stimulation, it will force all the nerves to fire action potentials at the same time. Again, that's a theory, and some surgeons, some are quite aggressive. They do cochlear implantation surgeries on patients with auditory neuropathy. For me, I want to be conservative. I'm not a big fan of doing cochlear implantation on AN patients. And for me, I'm not a big fan of that, especially if you want to make sure you don't implant on patients with normal behavioral thresholds. I mean, in reality, their hearing thresholds are normal. What's the rationale to do cochlear implantation surgery? And also, you do need to consult patients properly. Make sure they understand the pros and cons of the surgery. And if the patient has other neural involvement, if the patient coexists with other neural symptoms or other syndromes, that may not be a plus to do surgery. So again, this is the clinical profile for patients with autoimmune neuropathy. As you can see, it's quite messy, and the reason it's messy, maybe it actually contains a wide spectrum of different disorders. Because of that, there is difficult to pinpoint to anything very specific, because it may as well contain a group of different disorders. For the potential sites of lesion, first of all, we need to know that it's basically auditory dyssynchrony. It's whatever mechanism that will cause a neural dyssynchrony in the auditory system will result in expressions of auditory neuropathy. So any mechanism that disrupt neurosynchrony will create symptoms that look like auditory neuropathy as a result, as part of auditory neuropathy spectrum disorders. The possible sites of lesions are, say, the inner hair cell is the first one, and the synapse between inner hair cell and the auditory nerve, and the auditory nerve itself. It could be the dendrite, could be the axon, could be the cell body, could be the myelin sheath. It could be the neurons in the auditory brainstem area, could be in the pons or the midbrain area. It could be the efferent system, the so-called top-down mechanisms, because top-down mechanisms usually they provide some regulations, some inhibitions. Those will create different amount of delays. And again, there are many possible sites of lesions, and each site may just be a very different kind of disorder. We just don't know yet for our current knowledge. So again, there are many possible sites of lesion that may result in symptoms that look like auditory neuropathy. And as a result, they are now all considered as part of the auditory neuropathy spectrum disorders. So this can also be shown if you look at the medical history of patients with auditory neuropathy. It really contains a wide variety of reasons. The first one is no remarkable medical history. The patient just says everything looks normal, except they have symptoms that look like auditory neuropathy, or we should say they express symptoms of auditory neuropathy. Otherwise, we couldn't find anything wrong. Or patients with anoxia, they may have trouble during the delivery, the birth process. Or for some babies with hyperbilirubinemia, or the infants got infectious disease such as mumps, or some patient or children with immune disorders such as Guillain-Barré syndrome, or patients with some genetic or syndromic disorders listed here. There are a long list of genetic or syndromic disorders that can cause auditory neuropathy spectrum disorders. Some of these were permanent, some of these were transient, some of these were intermittent, such as high fever. For, say, a baby had a high fever when he or she was, say, 1 month old, and that could do it. Because, for example, the high fever, the high temperature may damage some structure of the myelin sheath, and those damage on the myelin sheath may be permanent or intermittent, and that will cause the propagation of action potential not reliable, particularly through the cell body of the auditory nerve. So it really could be anything. So it's again, it covers essentially everything. The most significant finding of ANSD is its effect on speech perception. This effect can range from relatively intact speech perception in quiet to the patient appearing functionally deaf. It's a wide range of performance in terms of speech perception in quiet. However, they all have extreme difficulties in noise. So regardless of whether their pure tone behavior thresholds are normal or functionally deaf, they all have extreme difficulties detecting sounds in noise. So again, this is the problem of neural dyssynchrony. So this is probably the common and most important finding for patients with auditory neuropathy. They all have extreme difficulties in speech perception in noise. Psychophysical testing results also show major problems in temporal cues. Again, temporal cues are related to the timing of the action potentials. So when the patients have trouble in timings of action potentials, they by default will have trouble in detecting temporal cues. For example, if you want them to do gap detection or binaural signal processing that involves timing differences in the, say, a gap in a sound or the timing differences between both ears, they will show extreme problems on those. Also, if you want them to do the frequency different alignments or intensity different alignments, they all have larger frequency and intensity different alignments when compared to normal hearing participants. So for speech perceptions, patients with ANSD, they have extreme difficulty in noise. They have problem in processing temporal cues. Their frequency and intensity difference alignments are bigger than normal people.